Hello students. Today we will be discussing about DNA. We all know that DNA is the genetic material. It is made up of a linear polymer of nucleotides. Okay. Now what are nucleotides and what it is made up of? So coming to the nucleotides, I would like to tell that these DNAs are responsible for the synthesis of RNA, and the RNA determines the protein. These proteins, which are formed from RNA, are responsible for providing structural and functional support to cells. Okay, so to construct a DNA, nucleotides are required. So, how these nucleotides are formed? So, the first thing which is required for the synthesis of nucleotide, okay, the synthesis of nucleotide is the nitrogenous base. Okay, what is it? It is nitrogenous base. So what are these nitrogenous base? These are nitrogen containing carbon ring. Okay, these are the nitrogen containing carbon ring. Now if we see, these nitrogen bases are of two types. Okay, they are purines and pyrimidines. Okay, these purines are double ring structure. Okay, they are having double ring, and these pyrimidines are single ring structure. So what are the bases which fall under purines? They are adenine and guanine. Okay, they are adenine and guanine. So the bases, pyrimidine bases, nitrogen bases are cytosine and thymine. Okay, but in case of RNA, instead of thymine, uracil is used. Okay, so adenine is represented as A, guanine is represented as G, cytosine as C, and thymine as T. Okay, so this is all about the nitrogenous base. Okay. Now, the second thing which is required for the formation of nucleotide is the pentose sugar. So what is this pentose sugar? This is a special type of sugar with five carbons. Okay, so this sugar contains five carbons. Okay, so this is the sugar. This is C1, C2, C3, C4, and C5. Okay, now when this two position, that is C2 and C3, has is having hydroxyl group in it. Okay, it is known as ribose sugar. Ribose sugar. Okay. Whereas when the hydroxyl group here gets substituted with H, okay, O is released. So it will become deoxyribose. Now this pentose sugar, these pentose sugar and nitrogen base fuses together to form nucleosides. So, pentose sugar, pentose sugar plus nitrogenous bases makes nucleosides. Okay. Suppose this is a pentose sugar, okay, containing adenine. This is a pentose sugar containing guanine. This is the pentose sugar containing cytosine, and this is the pentose sugar that contains thymine. So as they are fused together, so these are nucleosides. Okay. Now, when adenine is fused with the pentose sugar, it is known as what? Adenosine. Remember, adenosine. When guanine is attached with the or fused with the pentose sugar, it is known as guanosine. Okay. Now, when cytosine is attached with the pentose sugar, it is known as cytidine. And when thymine is attached with the pentose sugar, it is known as thymidine. Okay, it is known as thymidine. So these are all what? These are all nucleosides. But DNA is made up of nucleotides. Now, how this nucleoside forms nucleotides? When phosphate group is added to nucleoside, nucleotide is formed. Okay, so see, this is a nucleoside when attached with the phosphate. Okay, it will form what? Adenosine. Adenosine triphosphate. It is known as adenosine triphosphate. Okay, this is known as guanosine triphosphate. Okay, guanosine triphosphate. This is known as what? Thymidine triphosphate. Thymidine triphosphate. Okay, and this is known as what? Cytidine triphosphate. Okay? Now, as such, see, we see this is a this is adenosine, and as triphosphate is added to the molecule, it is known as adenosine triphosphate. Similarly, that is ATP. Similarly, this is known as what? GTP. Okay, this is known as thymidine. Thymidine triphosphate (TTP). Okay, and this is known as cytidine. Okay, now so the polymer of this nucleotide forms the DNA. Now, how this basic structure of DNA is formed? Suppose this is a nucleotide. This is a now the upcoming nucleotide which will join the first nucleotide. Okay, this is having three phosphate groups, okay, triphosphate. Now, the upcoming nucleotide loses two phosphate group, okay, two phosphate group to form phosphodiester bond, okay, with the first nucleotide. To form this bond, okay, this nucleotide has to lose these two phosphorus, which leads to the formation of phosphodiester bond. Okay, so this is a strong bond. Okay, similarly, the upcoming nucleotide, suppose this is the next nucleotide, it is T. So for this phosphodiester linkage, 2-phosphate is being released. Okay, so thus the backbone, the one strand, single strand of DNA is formed. Okay, now the two structures of the DNA are anti-parallel to each other. Okay, they are anti-parallel to each other and they are twisted. Okay, now we will see how these nucleotides are. Suppose this is a nucleotide. Okay, so this is the phosphate group 5 end. Okay, now suppose this is containing A. Now second phosphodiester bond O is containing C. Phosphate group O. This is containing suppose T. Again the phosphodiester bond. The next nucleotide it is forming G. Okay, so this is the 3' end and this is the 5' end. Okay, so the bases, so the base complementary to A is T, okay, and the base complementary to C is G. Okay, so these are the hydrogen bonds. Okay, so the new strand which is formed here is anti-parallel. 5' end. Okay, then the next nucleotide comes. Triple bond will be there. Next P, then again phosphorus. This is the phosphorester bond. Okay, this will contain G triple bond. Now P. Okay, this is the linkage, phosphorester linkage. This is again 3 end. See, this is the 5 end. This is the 3 end. And here, this is the 5 end. So these are the two strands of DNA. And these two strands of DNA, okay, remain in a twisted form in this fashion. Okay? Yeah, you have seen this diagram now. So this is the 5' end. So this will be 3' end and F. So this is the 5' end and this is okay. Now and in between them they are having what? Hydrogen bonds. These are the hydrogen bonds. So these are big bonds in comparison to the phosphodiester bond. Okay, these are weak bonds. So I want to mention one thing, that change in temperature or pH can break this bond. Okay, the bond between these nitrogenous bases. But this phosphodiester bond remains unaffected by temperature and pH. So, this was all about the structure of DNA. If you have any doubt or query regarding this video, you can mention below in the comment box. Thank you.